right, welcome back, everyone. I'm delighted to be chairing session 8 here. I'm Shana Kelly. I'm a member of the IIN and would really like you to welcome you to this next session. In this part of the symposium, we're going to celebrate the achievements of the winners of the 2021 Kabiller Prize and Awards. We have David Walt from Harvard, we have Professor Bin Liu from the National University of Singapore, and Professor Natalie Artzi from Harvard. These awards are given biennially, and the nominations are solicited worldwide. It's a very competitive pool of potential candidates, and the winners are chosen by an independent committee of world-renowned scientists spanning 3 different continents. These endowed awards are made possible by the generosity of a Northwestern trustee, alumnus, and chair of the IIN Executive Council, David Kabiller. So we'd like to kick things off and take a few minutes to show you a short video about David and why he created the Kabiller Prize and Awards.
Northwestern is such a special place. When I stepped on campus, there was an aesthetic to it, there was a Midwestern values to it, and it felt like home. Nanotechnology, it's really just understanding the building blocks of life. Years ago, the president, Henry Beanon, made a decision to say, let's really invest in this because it could change the world, and it's unfolding. It's multidisciplinary. People in chemistry, people in physics, people in material science, people in medicine. And so you need a place that has lots of different expertise, which Northwestern does, but also that people have the humility/desire to collaborate and break down those silos. When I look at the journey of where nanotechnology is, like, what does it mean? What's its potential? It's so complicated. I have a belief if we don't create understanding, we can't create engagement. I'm hoping with this prize that it's one small thing that we can do to create awareness and education. If I can help to apply my either energy or resources to be a catalyst to unleash the potential that's going on at Northwestern, I think it has the possibility of changing the world in so many positive, influential ways. And then we also wanted to create a Young Investigators Award because not only do we want to award lifetime achievement and importantly show education and impact, I also want to recognize younger talent, younger people who are seeing things differently and seeing what's possible, are not wed to the past. And so that's a really important aspect to the prize too, and to encourage others to follow in their footsteps. If we do the right thing and create the right culture of innovation and engagement, Northwestern has a real role in exponentially influencing the world.
Okay, so I'd now like to introduce the winner of the inaugural Kabiller Rising Star Award. Natalie Artzi is an assistant professor of medicine at Harvard Medical School. She's a principal research scientist at MIT's Institute for Medical Engineering and Science and an associate faculty member at the Wyss Institute of Harvard University. She has made extraordinary impact on the development of biomaterials for medical applications and has pioneered our understanding of tissue biomaterial interactions by demonstrating that the tissue microenvironment modifies or can modify an implanted material in a manner that is disease type and state dependent. This realization allows for the targeted, selective, and triggered release of therapeutics in a particular site and inside of particular cells. Natalie is a co-founder of BioDevac, a company that provides surgical solutions that aim to create materials that seal sutures post-surgery. Her work has been recognized with a number of awards, including the Bright Futures Prize from Brigham Health, the Young Investigator Award from the Controlled Release Society, one Brave Idea Award from Google Verily, AstraZeneca, and the American Heart Association, and as well, she has won the Women's Entrepreneurs Award from the Massachusetts Life Science Center. So let's give Natalie a warm welcome, and come on up, Natalie.
Thank you very much for the introduction, Shena, and thank you for the Kabiller organization for recognizing our work in the design of the materials for diagnosis and therapy. Congratulations, Chad, for the 20th inauguration of this center. It's incredible. This institute has generated such interesting work, research. I've had the opportunity to meet with a bunch of the speakers in the past few days. We found a lot of shared interest, made new friendships, and potentially new collaborations. So thank you for inviting me. So today I'm going to tell you about our approach, a new paradigm for the treatment of many hard-to-cure diseases that may affect each and every one of you. You may recognize some of the people here on the screen, and the idea is that it doesn't matter what kind of disease we have and how it manifests itself. There's a common thread between all of these diseases, which is the dysfunction of the immune system. Take brain cancer as an example, or alopecia, in which the body attacks its own cells, in this case hair follicles, that result in bald areas or sometimes becoming completely bald as a result of immune dysfunction. So this immune imbalance is a result of many of those diseases. And the idea of the immune system is that it needs to recognize threats to our bodies, respond to them, and eliminate them. But we see that this is not happening in many types of diseases. Here's an example in cancer. The immune system is supposed to identify the cancer cells, eliminate them, and in a way that will generate immune memory such that the cancer will not come back. And we know this is not always the case, which shows us that we need to develop new methods to eliminate cancer more specifically. In autoimmune diseases, like alopecia, for example, the immune system is hyperactivated, and in this case, attacking hair follicles. And what we want to do is actually suppress the immune system, but in a manner that's very specific that will help cure the disease. So immune therapy is a new paradigm by which we want to leverage our body's own immune system and reprogram it to eliminate the disease. In this case, what you see here is a T cell in our body that is recognizing cancer cells, attacking it, and eliminating it. And one of the major breakthroughs was the identification of checkpoint blockade molecules, those molecules that can help those T cells recognize the cancer cells by blocking the molecular brakes that sometimes prevent those T cells from attacking the cancer cells. So this new paradigm of leveraging the immune system is actually a different approach from the traditional methods, where we were looking for drugs to kill cancer cells, and which requires the therapy to reach the cancer cells wherever it is, in the pathological site as well as in metastatic lesions. But when we think about immune therapy, we're trying to generate a living and long-lasting response by reprogramming the immune cells, which means that the target as well as the release kinetics profile is not as clear, because while we need to reach the cancer cells in the primary tumor and in metastatic lesions, we may also need to reach some of the other secondary lymphoid organs, such as lymph nodes, spleen, and more. So my lab has developed multiple platforms for drug delivery that can help channel the drugs to wherever they need to go. Here, for example, you can see nanoparticles that can be injected systemically into our body, either intravenously, subcutaneously, or into the muscles in order to deliver cargoes. And you've heard a lot about nanotechnology this morning. The idea is to protect the cargo, shuttle it to wherever it needs to go, and enhance uptake into particular cells. But sometimes actually delivering those nanoparticles locally right next to the tumor imparts much better efficacy while lowering systemic toxicity. So my lab developed an adhesive hydrogel that can be injected right next to the tumor, and since it's adhesive, it will attach to it, and we can program the retention or how fast the material will degrade, which in turn drives drug release kinetics. So the material can interact reversibly with the drugs of interest, and that's why the release can be sustained and over time. Another local delivery approach is represented here by a microneedle platform we developed that enables proper delivery, transdermal delivery into the skin, enabling drugs to penetrate the skin by bypassing the stratum corneum, the outer layer of the skin that prevents many of the molecules from penetrating when applied topically. This platform is actually very easy to administer, is painless, and patients can apply it in the comfort of their own homes. So all of these drug delivery systems can enable us to deliver drugs where needed and modulate the pathways that we want and wherever we want. And why is this important? Take here, for example, the delivery of drugs that are given into the bloodstream or directly into the tumor here in the brain in a free form, or using our adhesive hydrogel that I told you about. You can see the distribution of the drug that is manifested here by the intensity of the color is very different in the brain, but also in the different organs. And it's not only the dose and accumulation of the drug in the different organs that will change, but also the duration. But here, look, for example, how the liver, lung, kidneys, and spleen light up significantly when we deliver drugs systemically. Now, when we look at the delivery and look at the time that the drug will accumulate or reside in a particular tissue, you can see here that following systemic delivery, the drug will be eliminated within a few hours. However, when we think about intratumoral delivery, drugs can be retained for a couple of days. And when we deliver the same drug and the same dose using our adhesive hydrogel, we can direct the release to last for up to 1 month, for example. So this has important implications in terms of the therapeutic outcomes that we will get when delivering particular drugs. This is an example of a nanoparticle we developed for systemic delivery of an immune adjuvant that you heard a lot about this morning, cyclic dinucleotide or STING agonist. STING is a stimulator of interferon gene, which activates many of our immune cells to recognize and attack cancer cells. So the nice thing about systemic delivery is that when we deliver drugs to the bloodstream, they can reach accessible as well as non-accessible pathological sites and metastatic lesions. However, a lot of the cargoes, when delivered into the bloodstream, will degrade prematurely. There will be not enough accumulation in the target site, and the uptake into cells is compromised. So we developed a nanoparticle, in this case polybeta aminoester. It's a polymeric material that is biocompatible. And we, for the first time, conjugated reversibly the STING agonist, or the CDN, which imparted much better serum stability, accumulation, and uptake into cells compared to when the CDN was electrostatically bound to the material. The polymeric chains were conjugated to this drug, to the CDN, using a cathepsin-sensitive linker, which means that when the particles go into the cells, cathepsin enzyme will cleave this link, enabling the drug to be released. Some of the other polymeric chains were modified with amino acids that are positively charged, and then they self-assemble by electrostatic interaction with the CDN to form this nanoparticle. And we wanted to see how well this nanoparticle works in vivo. And here you can see in a melanoma model in a mouse, when we deliver this CDN along with anti-PD-1 that you heard about, we see very nice improvement in mouse survival when we deliver the CDN, but this is much enhanced when we deliver the same drug and the same dose using the nanoparticle, because we can have many more copies of the CDN per particle. It's protected, it's shuttled to the tumor site, and the uptake into cells is very effective. Once we confirm that the CDN or the STING is the pathway that is really responsible to the therapeutic efficacy that we are seeing, we are very interested to see where the particles actually go following systemic administration. And you can see that, interestingly enough, in the spleen, we see major accumulation of the particles. And not only that, we see that those particles indeed activate a lot of the immune cells in the spleen, here dendritic cells or macrophages. And when we deliver the nanoparticle with anti-PD-1 compared to the free CDN, we see even enhanced activation. So we were very interested to understand the role of the potential role of the spleen in generating the anti-tumor efficacy that we are seeing, because this has huge implications for any immune therapy that we deliver systemically. So here you can see that we looked at the survival of mice that were treated with the CDN nanoparticles and anti-PD-1, either in a wild-type mice or mice that we resected the spleen. And you can see that for wild-type mice, this is the survival, and when we use the splenectomized mice, we see the same survival, but this is in the primary tumor. So this means how well can we eliminate the tumor? But the question is, what happens afterwards? Did the mice develop immune memory? Can they reject the tumor if we were to re-challenge them? So that's exactly what we've done. All cured mice were actually injected with an additional tumor, as you can see here, after 60 days. And what we found is that while the wild-type mice could reject the tumor, the splenectomized mice could not reject the tumor. And that was very interesting for us, realizing that the spleen actually plays an important role in determining the long-term efficacy of the therapy by generating immune memory. So while systemic nanoparticle delivery may work in many cases, there are diseases like brain cancer where systemic delivery is not going to allow us to get enough of the drug into the brain to reach therapeutic efficacy. And also, immune modulation in the periphery may not be enough to stimulate an anti-tumor immune response in the brain. So, in fact, our colleague and collaborator David Reardon at Dana-Farber recently in his talk just mentioned that one of the top reasons, actually 3 reasons, that a lot of the therapies failed and a lot of his clinical trials actually failed is because of lack of effective delivery systems. So here we turn to our adhesive material and wanted to see whether we can actually overcome this issue that, as you heard in this morning, most of the drugs cannot cross the blood-brain barrier. In fact, more than 98% of small molecules cannot penetrate the brain. So what if we were to deliver the drugs locally using our adhesive material? This material has a nice network of polymers that can interact with nanoparticles that you can see here in red that contain the drug and can deliver the drug over time. This is a platform that's easy to administer. Can provide with controlled delivery, and it's drug agnostic, so we can deliver any drug of interest. So how is this going to work? So the idea is following tumor resection in patients. We immediately will apply the material. The material is adhesive, so it will attach to the tissue and can release the drugs over time. It has mechanical properties that are similar to that of the tissue in the brain. So it makes it very biocompatible. So how does this help us? Now that we are actually right in the brain, we don't have to worry about the inability or the challenge of crossing the blood-brain barrier. So we can actually screen and select from a range of drugs that may actually be way more potent in eliminating the tumor, but could not have been delivered otherwise systemically, either because systemic toxicity or inability to cross the blood-brain barrier. So with that, we turn to initially chemotherapy that has cytotoxic effect, as I explained to you before. And this is very important in the context of glioblastoma. It's a very aggressive infiltrative tumor, and we want to make sure we debulk the tumor. But the nice aspect about doxorubicin is that it kills cancer cells in a way that it expresses molecules that can stimulate the immune system. And we were very excited about that and thought that it can potentially also synergize with the delivery of STING agonist that I told you about before that further activates the immune system. So here you can see the survival of mice. This is untreated mice. They will all die within 22 days. When we deliver the clinically used chemotherapy, temozolomide, we don't see any benefits. Now, when we use this much potent drug that we've identified, doxorubicin, that at least in vitro killed the cancer cells really effectively, we don't see a benefit either. And this is when we deliver the drug in a free form directly to the tumor. And the reason is that the drugs in the brain are being cleared very rapidly, so we don't see the effect. But when we take the same drug, same dose, and now deliver it using our adhesive hydrogel over time, we suddenly cure 60% of the mice. When we combine that with the systemically administered temozolomide, we now reach 80% survival of these mice. So this was very exciting. But we know that GBM really turns to recur. While it doesn't metastasize outside the brain, it does come back. So the question was, did we actually train the immune system using this drug that can express molecules that will educate the immune system what they need to attack? So to do that, here again, we re-challenge those mice after 60 days. All the cured mice, in the absence of additional therapy, were re-challenged with an additional tumor. And what we see is that 100% of them could reject the tumor. And we've actually mechanistically characterized the immune cells, and we've seen that indeed we've generated immune memory that enabled all these mice to be protected from this tumor. So what you can see here is that if we look at the tumor-draining lymph nodes, we see an increase in the generation of effector memory cells, which was, again, in the tumor-draining lymph nodes, so in the local regional effect. But we don't see, interestingly enough, any effect here in the spleen. So while following systemic delivery of immune therapy for the treatment of peripheral tumors, the spleen there played a significant role in generating immune memory, we see that in the brain it's actually different, and only in the tumor-draining lymph node we see the effect, so really local regional effect. That really motivates us to use local delivery system for immune modulation in the brain. Then we asked, can we actually combine the cytotoxic effect of doxorubicin that releases tumor antigens with the CDN nanoparticles, the STING agonists that we develop, that can present those antigens to T cells that then can kill the cancer cells. The other thing that the STING does is polarize macrophages that are 40% of the tumor mass in the brain, and polarize them into an activated phenotype that can clear the cancer cells as well. So we thought that this would be interesting to see if there's any additive or synergistic effect between these two treatments. So here you can see again the untreated mice that survive for 22 days. This is the doxorubicin when delivered using our hydrogel over time. This is the CDN nanoparticles alone. And now when we combine them, we actually see very nice synergistic effect. About 90% of the mice were completely cured. But we wanted to push it a bit further, so we threw in the anti-PD-1 that in many cases synergized with CDN, enabling those T cells to perform their action, and now we got 100% survival. Okay, so now we're good. We wanted to re-challenge these mice again to see if we generated immune memory, and indeed all of them rejected the tumor. So this was very exciting, really showing the strengths and importance of leveraging the right delivery systems, because even the same drugs, when delivered directly to the tumor, intratumorally inside the brain, we don't see the same effect. And this is really because we want to train the immune system and to bridge between the innate and adaptive immune system to generate memory, which requires a few weeks. And that's why we see those significant differences. Now I want to switch gears and explain to you why the local delivery is not only important to achieve efficacy, but actually it's important in the context of immune tolerance, when we want to actually suppress the immune system in the case of autoimmune diseases. And what you can see here, actually cannot see here very well, it doesn't project, but there are many T cells here that we're supposed to project, and some of them become autoreactive in autoimmune diseases. And the idea is that when we provide with systemic immune suppression, we're actually suppressing all the T cells in our body, really making our patients prone to infection, cancer, and other ailments because their immune system is simply down. We wanted to actually use a different approach. Rather than suppressing the entire immune system, we want to use immune regulatory approach in which we recruit T regulatory cells that can attack only the autoreactive T cells, so those that are pathologic, in order to restore immune balance, but leaving the rest of the immune system intact. And we wanted to see if in the context of alopecia areata, where the T cells attack hair follicle, resulting in these bold areas that you see, if we can modulate the immune system locally to restore immune balance, and how is this going to affect those patients, and what's the ability to potentially then protect them and leave their entire immune system intact. So the way we've done that is by developing a microneedle platform. These are materials with hundreds of polymeric projections in the micron scale that can penetrate the stratum corneum and deliver drugs effectively into the skin. Because of the micron scale, they actually do not reach nerves, so they're not painful and can be administered by the patients on their own. And this allows for delivery of a range of different molecules. So the idea was to apply those microneedles directly into the lesions that you can see, And we use two different types of molecules, CCL22 and IL-2, that can help with migration and proliferation of regulatory T cells. So we use the patient's own cells, we recruit them and expand them at the site of interest. The nice thing about those needles is that they can also sample biomarkers, so molecules and cells that reside in the skin, in the interstitial space. So we can also get information about the response to the therapy by actually retrieving those needles and cleaving the bonds, the polymeric chains here in order to get the biomarkers, and then we can analyze them, and this can help inform on personalized therapy based on what we see. And this will result in improved therapeutic outcomes. So that was the idea, and to do that we developed an alopecia model in a mouse, and the mice usually manifest with multiple lesions, as you can see here. And what we've done is we actually left some of the lesions untreated, and the one at the bottom was treated. We applied the microneedles, and you can see very nice hair growth where we applied the needles with the CCL22 and IL-2 that is also quantified here. So you can see in a score of 1 to 4 that 4 is actually complete hair growth. We can see that for untreated lesions we don't see a difference. Interestingly, when we deliver empty microneedles, because of the micro wounds, we do see some transient improvement. But then for the therapy-loaded needles, we see very nice recovery. Now the treatment was in the first 3 weeks. We stopped the treatment here, and you see that the effect is still maintained. And what we've seen is that indeed, if we take the skin and look for FOXP3, which is a biomarker for regulatory T cells, we see a nice increase for the treated skins, but we don't see any effect in the spleen, which is exactly what we wanted to achieve. We wanted to regulate the immune system at the lesion level, leaving the entire immune system intact to make sure that it can protect the patients. So what I showed you today is that materials can be designed to deliver a range of therapeutics to the right place at the right time and for the right duration. And this made all the difference in therapeutic efficacy and side effects that one could experience. With systemic delivery of nanoparticles, we can reach multiple sites and modulate the immune system in an organism-wide fashion. With the adhesive hydrogel, I showed you that we can modulate local disease and change local-regionally the immune responses. And with microneedles, I showed you that we can effectively deliver therapeutics and modulate the immune system locally to make sure that it's more safe and effective. And you've heard about the convergence today many times between material science, biology, and medicine, and we believe that this will help us really provide hope to patients and their families by delivering on the promise of immunotherapy using nanobiomaterials or their combinations. Thank you very much for your attention.
We have time for questions. If you'd like to ask a question, just signal for a microphone. It's been a long day. Microphone, and stand up just so we can see where the question is coming from. Questions? Chad?
Yeah, so that was fantastic. But if I look at the data for the GBM, where you ratchet it all the way up to 100% survival, which is remarkable. What's the biggest pushback? I mean, what or the biggest concern in terms of the movement from animals to patients? Because the data look extraordinary.
Yeah, we're always worried about translation from animals to patients. I think the encouraging factor in terms of sting is that when we take tumors from patients, there's actually a recent paper published in PNAS. They show that indeed sting is highly expressed in glioblastoma patients. So if we're worried if the target is the right target, it seems like this is the right one. There's also clinical trials now with doxorubicin. I met just yesterday with Adam here at the medical school. And I think one of the concerns with doxorubicin I had is, yes, it's a very potent molecule, but can also be toxic. And I was very happy to hear from him that in his clinical trial so far it seems safe. So it seems like the targets are expressed, the molecules are safe. So hopefully the combination would allow, and release over time, would allow us to translate that and see similar effect in patients. We're in fact starting a company in these days and working on raising funds. And so we believe in it and we hope it will translate to patients.
Exciting.
Thank you.
Thanks. Are those microneedles connected to a syringe? And does breakage of some of the needles, is that detrimental to the mice?
So the microneedles are actually part of a patch. So you can think about it as a Band-Aid that you simply apply to the skin. With a simple push, they penetrate. They have strong mechanical properties and their shape enables them to penetrate. We didn't see any adverse effect in the mice. We actually applied the needles every other day for a few weeks because we used it both for delivery, that we've delivered multiple times, as well as to sample biomarkers. So we applied also empty needles, let them absorb biomarkers, remove the platform, and analyze. So those mice have seen hundreds of microneedles within a few weeks, and they were fine.
Hi, thank you so much for that talk. Those two stories were very interesting. I also had a question about the microneedle project. I was curious how long-lasting that effect is if you're just treating locally, specifically if you're not affecting the systemic immune cells. Like, are you going to be needing to reapply the patch if the, I guess the space where no hair comes back?
Yeah, thank you for your question. So we believe actually that the effect will be long-lasting because we're actually training the T cells to identify those antigens and generate tolerance against them. So just like in cancer, we train the immune system to generate memory against those cancer cells, and we see a very nice rejection of the tumor upon re-challenge. Here we do the same effect. We train now the T cells to recognize antigens and create tolerance now rather than activating them against those antigens. And we've seen some of these effects by stopping the treatment at week 3, and now some of these mice are week 20, and we don't see any relapse. So it seems to be robust for now.
Hi, really cool, and congratulations as well. I'm curious if you can talk a little bit more about the chemical structure of the hydrogel, as well as what's affecting kind of like the release kinetics of the drugs in this case. Thank you.
Sure. So the hydrogel is based on 2 polymers, dendrimer, polyamidoamine. We use generation 5. It has 132 molecules that we can block some, we can modulate some. And we have dextran as another component that we oxidize to form aldehydes. So these amines on the dendrimer and aldehydes in the dextran form spontaneous reaction. So we wanted to make sure that we can have a material that will gel immediately and does not need light, curing enzymes, or any activators. So this is really easy to use. And the other nice thing is that this bond that is formed, this imine bond, is reversible. So it will undergo hydrolysis over time. So the material will degrade and you don't need to remove it. And what will control the release kinetics of drugs is actually the cross-linking density or the formulation of the material. But beyond that, actually this material has a lot of excess functional groups by design. Those functional groups can reversibly interact with drugs, which means that the drugs will be released as the material degrades. And since we can control the degradation of the material, we can program it to degrade within days, weeks, or months. Drug will follow the same trend. So that's one of the features of this material that enable us to release a range of therapeutics over a few weeks.
Okay, we can take one more question if there is one. Yeah.
Hi, thank you for the great talk. I guess kind of going off the other gentleman's question, is the importance of using the patient's own cells for like the microneedle project, is that for the specific training of the T cells, or what's the importance of using the patient's own cells?
Yeah, so I think using the patient's own cells is the easier approach. Also from a regulatory perspective, If you need to transfer cells, which is something we also do, in some of the models, it's a bit more complicated. Also, if you need to take the cells, remove them from the patient's body, modulate them, and reinfuse, it makes it a bit more expensive, time-consuming. From a regulatory perspective, is more demanding. So if we can deliver a therapeutic locally, attract the cells of interest, and reprogram them at the site, that's the easiest approach. But of course, in cases that it doesn't work, one can consider also T-cell transfer as well.
Okay, well, I think with that, let's congratulate Natalie again and thank her for a terrific talk.
Thank you. Also, Professor Artzi, what a wonderful talk today. I really enjoyed it as well. Folks, my name is Ted Sargent, and I have the pleasure of introducing the winner of the Kabiller Young Investigator Award, Professor Bin Liu. Professor Liu is unfortunately not able to join us today, but we would like to honor her many contributions. Bin Liu is a distinguished professor and senior vice provost at NUS, the National University of Singapore. She's co-founder of Luminicell, a Singapore-based biotech. It's focused on bringing next-generation cell imaging contrast agents to the market. She earned her bachelor and master's at Nanjing University. She finished her PhD at NUS. She was a postdoc at UCSB, and then she returned to NUS in 2005 as a faculty member. Professor Liu has made outstanding contributions in the development of platforms that advance biomedical research and nanomedicine. Her invention of bio-nanoprobes enables the visualization of cancer metastasis and the monitoring of tissue regeneration after stem cell transplantation, rapid disease diagnosis, and drug screening. Her team has integrated therapeutic functions into organic molecules to develop novel platforms that combine image-guided cancer surgery with treatments. Most recently, the signal amplification technologies developed in her lab were used for COVID-19 protein detection. Please join with me today in honoring Professor Bin Liu.
Okay, well, it's been an exciting day of great talks, great discussion and getting together. And it's time to introduce this year's Kabiller Prize winner. So it's really my pleasure to introduce a longtime friend, collaborator and colleague in a number of outside activities, and somebody who is extremely well-deserving of the Kabiller Prize. And this is Professor David Walt. You can applaud in a moment. I'm going to say a few more things. David is the Hans-Georg Wyss Professor of Biologically Inspired Engineering at Harvard Medical School in the Department of Pathology at Brigham and Young Women's Hospital. He's a member of the Wyss Institute for Biologically Inspired Engineering, and he's also a Howard Hughes Medical Institute professor. David's career from his early days have been really in the role of pioneering bioanalytical chemistry and more specifically optical-based sensing and sensors and sensor arrays. And I still remember the paper he published in the late 90s that described a really ingenious way of self-assembling biomolecular arrays, assembling beads in wells, dimples on a plate, so that you had an array of peptides or DNA, but you didn't know what sequence was in what position. And he came up with a clever way of decoding the array. That step, which was like not like anything before it, led to the creation, the founding of Illumina, what today everybody recognizes as the global leading company for providing genomics tools. And I think anybody that's done an experiment in genomics has used technology that's come from Illumina. And so David's name is really synonymous with optical sensing, biotechnology, and ultrasensitive assays. After Illumina, he had another really clever and unique idea of how to develop systems for ultrasensitive detection down to single copies of proteins. That work, which he's termed Simoa, also led to the founding of an exciting company, Quanterix, and a company that's now also publicly traded. Between the two of these, there's a market capitalization that's reached well over $60 billion, so that's an enormous impact that the science has had. And these tools have had even more impact. They were foundational during the initial development of the COVID vaccines. They were used to identify viral targets and really became part of the Pfizer, Moderna, and J&J approaches to the vaccines. And so you can imagine the creativity and insight here, how big of an impact and reach it's had in making our lives better. Professor Walt has received, as you would expect, numerous honors and distinctions and recognitions. I'll mention his elections to the National Academy of Engineering, the National Academy of Medicine, the National Academy of Inventors, induction in the Inventors Hall of Fame, and a number of ACS awards, including the Catherine Haack Award for entrepreneurial success. So before we bring David up, I'd like to share a short video that describes him and his work.
I grew up in Detroit, Michigan. I primarily spent my time riding my bike and going out exploring unexplored fields and forests. I first got interested in science through these explorations that I took, but I knew that I wanted to be a scientist as a profession when I was in my first year of college and joined a research lab and just started to make things that nobody had ever made before. It really gave me a thrill to recognize that in the whole history of the world, I was the first one that was now encountering this material. After my graduate school training and postdoctoral studies, I fairly quickly got into the field of sensors and biosensors. A number of collaborators to my lab, they said, we'd love to measure 100 or even 1,000 things at a time. And at that moment, I had what I think could only be described as an epiphany. We could create these very high density microarrays that had unlimited capacity to do a huge amount of genetic analysis. Microwell arrays were taken and used to create very, very dense nanowells that enabled the human genome to go below $1,000. 10 years after that initial discovery of microarrays, we applied that to the measurement of proteins. And so we invented this technology called Simoa, which stands for single molecule arrays, that allows you to detect proteins at extremely low levels by creating a fluorescent signal that sort of shined like a supernova. You now have the opportunity to detect things at much lower concentrations. Nanomedicine is an entirely new field that enables new forms of diagnostics, new forms of therapeutics that have the ability to, I think, transform healthcare. We're really focused on trying to get people much earlier in the pipeline interested in invention.
We might miss some.
One of the most delightful aspects of my career is having hosted so many bright, inquisitive, creative people, and then watching how they apply that after they leave to their own careers. When I received a call that I was the winner of the 2021 Kabiller Prize, I was speechless. The award highlights the future potential of this field to really make a huge difference in the world. When we wake up as scientists, every day we don't know what new thing we're going to learn. The discoveries that you make may in fact have the potential to transform lives. We have to keep trying because those are incredibly important problems to solve.
Let's welcome up the Kabiller Prize winner in nanoscience and nanomedicine.
Well, thank you, Milan. Just watching this video myself, I realized it was taken a little bit of time ago with a little less hair up here. Maybe Natalie, you can help out with that. So first of all, everybody's congratulated Chad today, but wow, I mean, you've really created something here that's special, Chad. You know, this is a fairly insular group because most of you are from Northwestern. But from the outside, this is the place for nanoscience and nanotechnology. So, you know, you should be proud of what you have here because it really is the world-class place where nanoscience and nanotechnology, nanomedicine are being done. So congratulations, Chad, not for just, you know, establishing this, but just for all the amazing things that have arisen as a consequence of having be here. So John Rogers earlier today made the analogy, said he was the appetizer, and then I think he said Steve Shu was going to be the main course. Well, I'd like to think of this as the dessert. But it's a prefix meal. So you don't get a choice of what you're going to get served here. But I can assure you that it's sort of the low-calorie version because I'm not going to talk a lot about technology or science and get deeply into it as the last talk of the day. And so you saw what I'm going to talk about: discovery, scale, and impact. And you saw a few flashes of some slides that I'm going to go into a little more depth in as I go through the talk today. So first, discovery. And I'm going to tell two stories here. I'm going to tell the Illumina story, and I'm going to tell the Quanterix story. And both of them have a discovery phase, they have a scale phase, and then they have an impact phase. And so as Milan described earlier, this was sort of the substrate that we worked with. This is an optical fiber array. This was something that was created at Corning Glassworks. It has, you know, at this stage of the game, it had about 2,000 fibers in this particular array. And we are trying to accomplish something that I'll show you in a slide, in two slides, but this is what happened. Every time we treated this glass fiber array with hydrofluoric acid, dilute hydrofluoric acid, the cores of the fibers in this array etched a little bit faster than the cladding material. We kept getting these wells, and that was not what we wanted to get. We ended up publishing a paper, I think it was 1996, it says here, and you can see we made micro well arrays, we made nano well arrays. So, you know, this was 1996, right around, you know, the time you guys were thinking about nano as well. And this was exactly the opposite of what we were trying to accomplish. We were trying to make these nano tip arrays. I'm not going to tell you why we were trying to do this. But this is what we were trying to do. And eventually we were able to do this. But you see that this was the year after those micro-well arrays were published, but we had no concept, no idea what they were good for. But this is where we get to scale. If you think about what we had accomplished, or if I had thought about what we had accomplished when we actually published this paper and made these little micro wells, you can see that there's a lot of zeros on this slide here. 1,000 times as many wells in that little fiber that I'm holding in my younger hand at the time, 60,000 of them, in fact, compared to what was considered a microwell array back in the day, which was the 96-well microtiter plate that we all use for biological experiments. And so that's 5 billion times smaller, okay? And I always try to encourage, particularly the students in the back of the room, because for some reason you always stay back there. If you ever see that many zeros, pay attention to it, because we didn't do it. We didn't do it for about 3 years after we had published that paper in Chemistry and Materials. But over time, what happened is the fibers got better. The Corning glass was making better fibers. Others got into the field of optical fiber arrays. And a key experiment was done by a graduate student in the lab, Carrie Michael, just for fun. She took some latex beads and she said, gee, I wonder if I could put those into those little wells. And she took a little droplet of a slurry of those little latex beads and put it on top of that microwell array that I showed you I was holding in my hand a few minutes ago, and took a Kim wipe and wiped it off, and then took a microscope image, and lo and behold, these little beads fit into each of these wells. They just nestled their way in, just a true self-assembly process. Well, again, we had no idea what this was good for. I mean, this was not a deliberate sort of focused effort to develop a new technology. This was just an observation that somehow I filed away. Well, it turned out, as Milan articulated a little bit in the introduction, that what we did was we created this thing called random arrays. And I'm not going to go into the details of how we did this, but what this allowed us to do is it allowed us to put these beads in wells with either peptide sequences or DNA sequences, and we could decode them so that we could create these arrays randomly. Every array was different in terms of the positions of the beads, but every array had the same library of beads. We just had to figure out where the relative positions were of each of those beads after we made it, and that became the basis for Illumina. And when we first started doing this with nucleic acids, this was the smallest size of a feature in one of these, what were called gene chips at the time, that were made with a fairly arduous photolithographic process by a company that some of you people will remember, but it's sort of been relegated to ancient history called Affymetrix. And this company was sort of the leader in genetic analysis at the time, and we could now pack many more pieces of information in the same real estate as they could using this rather complicated and expensive process. And so we started this company called Illumina, which has become, I think, pretty much a household name. Now, I was told that it's a pretty mixed audience here, so let me just spend a minute talking about nucleic acids and DNA. You all know that there's about 3 billion bases in the human genome. And of those 3 billion bases, all of us in this room, each of us differs by 1 in 1,000 of those bases. So 3 million bases are commonly what constitutes the entirety of human variation on the planet, okay? 1 in 1,000. So we need to measure, if we want to completely capture most of the common regions of variation, these different bases in the human genome. But it's not a random process in the sense that each of us has 3 million distinct bases. It's, see, Natalie, I said one of us should be able to get this without going forward or backwards. She did it. I did not. Okay, so let's focus right here at this A and G. And let me go over to this side so I don't have to crane my neck. So this is one human right here. This is another human right here. And at this particular location, everything else is the same for 999 bases. But at one particular location, some of you in the room will have a G at that location, and some of you will have an A. And the frequency of each of these variations might be 50-50, it might be 80-20, it might be 90-10 in terms of what the statistics are at a particular location, okay? But each of us will have 3 million of these bases, and that's what defines how we're different from each other, by and large, okay? And those are called SNPs, single nucleotide polymorphisms. So those SNPs, single nucleotide polymorphisms, constitute pretty much the entirety of human variation. Now, at the time that we founded Illumina, it cost $2 to measure one SNP, okay? So if you wanted to measure one person's entirety of variation, it would cost 2 times 3 million, $6 million to do that. That's just economically not feasible. So we came up with the bead arrays, which completely transformed the whole process. When Illumina first introduced their product, so the company was founded in 1998, first product that was introduced in about 2002, 2003, it was an array of 96 of those fiber optic bundles with beads located at the end of them, and it just dipped into a 96 well plate. So you could do 1,536 SNPs on each of those fibers times 96. So that was, whatever the number is here, roughly, a few 100,000 SNPs in this, what we called an array matrix. And then eventually that just got to be both arduous to make as well as too expensive. And so things migrated onto just etched silicon chips. And you can see what happened over time is this past year, 28 million markers per chip. 28 million markers per chip. Now, I just told you that pretty much the full complement of human variation is 3 million bases. Why 28 million? Well, it turns out that there are rare SNPs that lead to different diseases. And so as sequencing has gotten cheaper and more people get sequenced, we find more and more rare SNPs, and those can just be migrated onto this array format. Now, the reason that in 2021 there are still these bead arrays being sold in millions of quantities is because it's so much cheaper to measure 28 million SNPs than to sequence a human genome, okay? It costs less than $50 to measure 28 million SNPs today. $2 a SNP back in 1998. This is what that chip looks like today. I just got these from former student who's now vice president of chemistry at Illumina. So in this little location right here, 28 million beads in wells. How do you measure 28 million? You have to measure 28 million times 30 because you want to make sure that there's a representation of every one of those 28 million SNPs in that array. You can't just put 28 million beads in, or many of those, statistically, you'll miss a lot of those sequences. So you have to have an overrepresentation of the number. So it's not just 28 million beads in that spot, it's 900 million beads in one of these little wells. So there's wells in wells here. So what do those things look like? That's what they look like right there. Little bead snuggled into one of these wells. This is, you can see we heard about diffraction patterns due to nanostructures earlier today. So $2 goes to 4 millionths of a cent. 4 millionths of a dollar. Okay? 4 millionths of a dollar. That's 500,000 times cheaper. So that's how it can be done. And what's done with these? Well, they're used for lots of things. This is one of the biggest uses, not quite the biggest use, 23andMe. They don't sequence. You send your saliva sample in, and they run one of these bead arrays on an Illumina chip. And there are 12 million participants in 23andMe at this point. 23andMe, it's like the people who do the recycling, the plastics recycling. They charge the city to cart the plastics away, and then they put it into some form, and then they sell it. So they get paid both ways. 23andMe is sort of like that, kind of like the recycling collectors, because they charge you to run your genome, and then they aggregate all the data that they get, and then they sell it to pharma. Okay? But it's the biggest, what's called genome-wide association study that's ever been done, because they have 12 million people that they've collected genetic data on. And it's a rich, deep database that, that they can mine, and they ask people, can you taste, do you like broccoli? Can you smell asparagus, after it gets peed out? These kinds of interesting recreational questions. But these are things people are interested in. But for free, you also get your maternal ancestry, you get your paternal ancestry, you connect with people, who are genetically related to you. I get emails. I was one of the first 100 customers, and I still get emails from DNA relatives, that are like third or fourth cousins. I don't even connect with second cousins. So this is the biggest use, Ancestry.com. There's 20 million users of Ancestry right now. So, people, 12 million people are interested in sort of some of their health traits and other things. Everybody's interested in their ancestry. It's sort of a free family tree, right? You don't, at some point, probably in 10 years, you won't even have to keep track of a family tree, because you just go on to a website like this, or you go to 23andMe, and they'll just make the connections for you and tell you who your great-grandfather and great-grandmother was, and which country you came from. So Illumina also was instrumental in the Human Genome Project. And what's shown on this side right here is just one of 40 institutions. I mean, these were huge labs running initially slab gels. Eventually, Lee Hood developed capillary sequencing that was used later in the Human Genome Project. But pretty much what was done was 1,000 bases at a time were measured on one of these, gels. It took 13 years. You got one genome. They did it 8 to 9 times. So what they did is, just because of the errors that are intrinsic to doing sequencing, you have to do multiple experiments to make sure that you get the right letter, the right base in the right location. So that was done at what's called a coverage of 8 to 9x. It means it was sequenced 8 to 9 times, and then if there was an error, they would pick it up because 1 out of 8 or 2 out of 8 would give you an A, and the remaining ones would give you a G, and then they'd say, well, there must be a G at that location. And it cost a total of $3.8 billion for the project, okay, for the project. Now today, actually not today, but last month, Illumina announced a NovaSeq instrument that does 16 trillion bases on one instrument in one day. That's with one full-time equivalent employee. That means 200 genomes at 30x coverage at $200 per genome. So what does that mean? That's the entire output of the 13-year Human Genome Project in 7 minutes. 19 million times cheaper and 1 million times faster than 40 labs combined. This was done from 1990 to 2000. Wasn't that long ago, right? Never in the history has technology developed this fast in any field. This is, the latest that was put out by the NIH, but they haven't gotten to the point where they lowered it from about $800 to $200, which is now the new price of the sequence if you run it on one of these high throughput instruments. Few interesting features about this slide. Clearly, this technology, when next-gen sequencing was introduced, it just completely outpaced Moore's law. And in fact, for a while, right around here, it got more expensive to store the data than it did to do the sequence. And then they decided that sequencing was cheap enough that they didn't want to store it anymore. It was just, if you made a mistake, you would just resequence it because it got to be cheaper than storing the data. The other thing I want to point out here is that sequencing was done, and I'm not going to, again, go into the details, with something called synthesis by sequencing by synthesis, and it got stuck at about somewhere between $5,000 and $10,000 for a genome. And it was stuck there for actually quite a while, for a number of years. There wasn't any progress here. This is pretty flat. And then what happened was Illumina had the idea that let's migrate our sequencing chemistry onto those microwell arrays that we have. And what that allowed them to do is it allowed them to dramatically increase the density and dramatically decrease the cost. And that's when they achieved the $1,000 genome. It was a step change. You see, it went from about $5,000 to pretty close to $1,000 at that point. So, this is important. These figures are important in terms of market cap and all that. And market has dropped in all sectors. So, I think the $65 billion, I think it went up to $90 billion at one point, but it's back down in the about $40 billion range. Has, about 11,000 employees, globally, $4.7 billion in revenues last year, and a lot of awards here. But I want to point out that this is a team effort. Okay, scaling at this level requires brilliant, and we're here in Kellogg, brilliant business people, financial people that can really help secure the kind of cash resources that are required to capitalize the company at the right time in the right way. It requires incredible innovation in sales and marketing, engineering, science, biology, everything. So it's a true team effort. And for me to be standing here, there's a huge team that it took to really bring this to fruition. And others mentioned the issue of translation, whether it's to the clinic or to the commercial sector. It really requires collaboration and teamwork. Notice I put all that stuff before impact, because one could say, well, creating 11,000 jobs, that's pretty impactful. I'd say that's true, it is. But where the real impact comes to play is here. So on the left-hand side, you see Carmen, the mother, and this is Lucas, this beautiful baby boy. So she suffers from a rare disease called Charcot-Marie-Tooth syndrome. It's a debilitating neurodegenerative disease, muscular wasting. Her grandfather had it, she has it. So they did pre-implantation genetic analysis using our technology. So in vitro fertilization, they take father's sperm, mother's harvested eggs, fertilize the egg, start growing embryos. And then they take some of those cells, amplify them, and genotype them using our beta rays. They can then select the embryo for implantation that does not contain the genetic defect. And that's how Lucas was conceived and born. This family will not have the risk of Charcot-Marie-Tooth syndrome ever again, because it's been taken out of the germline. Natural DNA from the mother, natural DNA from the father. It's just, usually it's the luck of the draw, right? Here, they took the risk out and took that horrible disease out of the family line. The picture on the right, different story. When this technology was first introduced into clinical use, what was done with these arrays was, one of the things that was done was to actually look at the intensity of some of the genes that were being measured. And it was being used actually to measure, using non-invasive prenatal testing. You could take the mother's blood and you could actually see the fetal DNA that's circulating in the mother's blood, and you could look at the ratio of chromosome 21 from the mother and from the fetus. And if it was 1.5, meaning two chromosomes from the mother, 3 from the baby, that indicates trisomy. That's Down syndrome. Okay, and that's what these tests were being used for. And the mother, her name is Marin Mahia. She had this done. She was 39 years old at the time. This was a common test that was being done. It still is being done now. It's being done primarily through sequencing. But what it turned out was that there was some chromosomal anomalies in the bloodstream. Well, it turned out that this was one of the first cases where they found circulating cancer DNA in the maternal blood because of the abnormal chromosomes that were circulating, and they were maternal chromosomes, so maternal DNA. And so what they did is they delivered the baby early at 32 weeks so that she could undergo chemotherapy, because chemotherapy is not good for a fetus. But this is impact. This is clinical impact. And this is why we do the things that we do. And we heard so many other kinds of stories today. So the second story, not going to take as long, the discovery was actually on the same platform. It was using these microwell arrays that we had created, but it involved a very different kind of approach. And this approach was that we recognized, it was actually at a lab meeting, we just did the calculation. You know, if you take one of these little microwell arrays right here and you calculate the volume in one of these little microwells, it turned out to be on the order of a few femtoliters, okay? 10 to the minus 15th liters. That's really tiny volume. And then we said, well, how many molecules of a fluorescent dye would you need to put into one of those little wells in order to get a signal that you could see with just a regular microscope? And it turned out to be, you do the calculation, just a general chemistry calculation. Turns out, you know, 100 molecules, okay, so that gets you up to a nanomolar concentration, pretty easy to see that. And sure enough, you know, we sort of bias the system in our favor by putting in an enzyme with millions of molecules of a substrate molecule that got converted into a fluorescent product. But you can see that, you know, here's background where we have the substrate molecule but no enzyme. And then if we have one molecule of enzyme in each of these every 5 wells, you can see these things lighting up quite brightly. And then, you know, as we dilute, you get fewer and fewer wells. And so this was really the first experiment that was done where you could actually measure the concentration of something by counting the number of molecules. I mean, other people had counted molecules, you know, in sort of the, you know, fluorescence correlation methods, but this was the first time you could just look and count molecules and determine a concentration. And so, you know, this is just for, you know, some people in the audience who aren't biologists. You know, DNA gets transcribed into RNA, gets translated into protein. This is the central dogma of biology. We said, you know, one thing that's really tough to measure, we can measure any gene we want now, we can sequence things, but we can't measure proteins that well. So that was the next challenge. And the reason it became a challenge is because if you look at the number of approved tests, clinical tests for proteins that have been approved by the FDA that are clinically meaningful, there's 171 of those. And those are defined by a concentration that's, you know, again, I'm not going to try to be technical here, but that's picomolar concentration and above, okay, 10 to the minus 12th molar and above. Not sort of really ultra sensitive. But if you ask the question based on all that we know from the Human Genome Project, how many proteins should there be in the blood? It's thousands. And we know now from, you know, Neil's work and others, you know, that you know, are doing mass spec, that there are thousands of these proteins in the bloodstream. But what was a problem was that you just couldn't measure them with the technology that existed at the time that we thought about this problem. And so what we decided to do was to convert the way that things were being measured, proteins were being measured, from what we called an analog signal, meaning just an intensity change in a microtiter plate or, you know, on a piece of paper, to one that was digital, because this is an absolute measurement. You can tell that there's 4 molecules here. Okay, absolute measurement. You don't need to calibrate. So we now published a paper. What was this? 2007, something like 2010. And then, you know, shortly before this paper came out, founded a company called Quanterix, which, not in San Diego, but in Cambridge, Massachusetts. These are the instruments that are used to carry out these studies. And then let me just talk about scale here, because, you know, the fibers didn't work for this. So we looked for a platform. It was not a silicon-based platform. it turned out here that this was a plastic material that was in search of an application. I think some of you gray-haired people recognize, this format. These are used to be called DVDs or Blu-rays. Nobody uses these things anymore. But we engaged with Sony and said, you guys have this technology that can make all sorts of nice micro and nano scale patterns on these plastics through imprinting. Could you make some of these micro well arrays for us? And sure enough, they created these little channels here that allowed us to load our samples with, again, I'm not going to go into the technology in detail, but I think you saw a flash of a picture here that was done by a scientific imager, Felice Frankel. I think she's probably made her way through IIN at some point or other. But the arrays here are 216,000 little wells here that get loaded with immunocomplexes that signal the presence or absence of a protein molecule. And this is what they look like. This is what the images look like. And this is what the beads look like. And you notice these are roughly a micron-sized beads. But the key to the success of this assay is, we heard it before, the high surface area. The nanostructure on these beads is critical to their performance. If these beads were just smooth, wouldn't work, does not work. So this is a critical component to these to the performance of these single molecule assays. And again, you can see that we've created these. These can measure multiple proteins simultaneously, and they're much more sensitive than the standard type of assay. So the company, again, market cap, hit like 2.5 billion, and it still has 500 employees, and last year I think 120 million in revenues. I'm a director of the company. It's moved out to the Boston suburbs at this place just because real estate's so expensive, and did an IPO in 2017. I'm just going to close with a few other types of impact here. So this is the course of a patient who has been diagnosed with prostate cancer. They usually get diagnosed because first their PSA level, prostate-specific antigen level, is high, blood test, and then if it turns out to be aggressive or the patient turns out to be relatively young, most men will die with prostate cancer. They don't die of prostate cancer, but if they're diagnosed young, then they may want to resect the prostate. And when they do that, it's called a radical prostatectomy. What happens is right after surgery, the PSA level plummets to below the detectable level. And so I'm sure there's many men in the audience who have had friends or family relatives, men or women, who've had who have relatives who've gone through this type of surgery. And what happens is after the surgery, the patient has to come in every 6 months to get tested, because if there was residual tumor left in during the surgical resection, or the tumor had metastasized prior to the surgery, then what will happen is that residual cancer that's left in the body will grow, and over time it grows to a point where now the PSA level gets to where it can be measured again. And so what the physician will say upon a consultation is say, you have biochemical recurrence. What does that mean? It means that we have a really crappy test that can't measure what needs to be measured because it's not sensitive enough. So what we did is we converted this PSA test into a Simoa test, just this digital assay that we created. And we took samples from men who had unmeasurable levels of PSA after 5 years after their surgery. And this is what we measured. We could quantify every one of them. The test is 3,000 times more sensitive than the existing test that can measure that can measure PSA. And so now we can quantify these patients. And we then took some retrospective samples. These were samples of patients that had had the surgery, and then they were collected at various time points. And what we could do is if we took a single time point between 6 weeks and 12 weeks after their surgery, and those patients had a PSA level of less than 3 picograms per mil, which was not measurable prior to this technology, 100% of them had no recurrence after 5 years. But if it was greater than or equal to 3 picograms per mil, then they only had a 35% chance of being cancer-free after 5 years. And so now this test is being used, not just a single time point, but you can actually measure people, and if their levels are stable, then it means that they don't have any residual disease. If in fact things begin to go up, they can intervene earlier than waiting for things to pop up above that level that previously was considered biochemical recurrence. This is really the last slide, and I just want to describe briefly where this technology is now having an impact. Its biggest impact right now is in neurodegenerative disease. There's a protein marker that was discovered using this technology called neurofilament light. It's a protein that's released from nerves that are damaged, and those nerves can be in the brain. They could be they could be systemic, but any nerve damage will cause this protein called neurofilament light, probably appropriately abbreviated NFL because of concussion, but it's now been approved, has breakthrough designation. This is a breakthrough designation run in a CLIA lab at Quanterix for multiple sclerosis. So it's a great predictor of progression of multiple sclerosis, as well as the efficacy of drugs that are designed for these patients. The other place that it just got breakthrough designation is a protein molecule called phosphotau 181. So it's a serine residue in the tau protein that is implicated in Alzheimer's disease. And if you have abnormal phosphorylation of tau, and it occurs primarily at position 181, so this is a particular modification of this protein, you get these tangles, these sort of fibrils in the brain, and it's being used for both diagnosing Alzheimer's disease, but also being used in dozens of clinical trials that are being used with all these companies that are now coming out with Alzheimer's drugs or drugs that putatively will be designed to be efficacious against Alzheimer's. And so stay tuned there. My lab's focused on early diagnosis of Parkinson's as well as Alzheimer's. We're also working on a combined liquid biopsy using this technology. We have a panel of 20 protein markers for early detection of breast cancer. Our initial focus in this is really to combine mammography, because that's just the way that screening is done for breast cancer, with this blood test to eliminate 80% of all the biopsies that get done. Because 80% of all the biopsies that are performed on suspected breast cancer turn out to be benign. And so if we can eliminate that, then we can, I think, really make a difference. And the reason that this works is because our technology is sensitive enough to detect a tumor that is really tiny compared to what you can see with an image. Imaging, the best imaging is able to look at 0.5 centimeter, 5 millimeters. So if you do the math, that's 125 times smaller volume, which really would be a much earlier stage cancer than if it gets to 0.5 millimeter. And we're also, we've also have Gates Foundation funding for both tuberculosis and HIV, and recently published a paper that got 70,000 downloads and I don't know how many tweets, because we found the spike protein from the virus in 65% of the long COVID patients. So this is really the first real biomarker that suggests that there is a persistent viral infection in these patients who are suffering from long COVID, and there's millions of these patients. So we're now initiating a clinical trial to give Paxlovid for 15 days. It's approved. Safety profile, it's in the phase 0 study, phase 1 study, it was done 15 days. So Pfizer is sponsoring a study at 100 sites across the country, and we've transferred this to the clinical laboratory to be the reference lab for the entire national effort. So these are people, both not present people, but all the people who sort of were instrumental in developing some of the technologies that I talked about today. The funding agencies. This is a more recent picture of the lab, but I want to both express my deep appreciation to David Kabiller, to the IIN, to Northwestern, to the selection committee. A true and real honor. One of the things I think that was most striking to me today in listening to all these spectacular talks is that there's probably a decade of Kabiller Prize winners just here at Northwestern. It's probably politically inappropriate to give it only to Northwestern faculty and scientists, but a lot of deserving people really who have done incredible work. So thank you very much. Again, discovery, small piece, scaling, big piece of the translational puzzle, and then really the reward is when you begin to see these technologies really have the impact that they have, not just on new discoveries, but on patient care and unmet health needs. So thank you very much.
Great. David, that was a tour de force ×2, and you get your reward now. And because no one wants to see you drink alone, I'll join you. Cheers. Cheers. Okay, we've got time for a few questions. Chad.
It's absolutely tour de force. I think that's fantastic. You look at the Illumina story. I mean, I did not understand a lot of the details of it, but I mean, prior to this talk. But I think you said that the original beta rays or versions of them are the primary component of 23andMe. Is that correct? And Ancestry.com, yeah.
Ancestry, yes.
Yeah.
And National Geographic.
So if you break it down, I mean, so everybody focuses on the sequencing. What percentage of Illumina's current business is the SNPs via beta rays versus sequencing?
So it's interesting. Probably, I've been off the board for a little over 5 years now. So it's all a guess at this point. But I would
just rough.
Yeah, I would say of the 4.7 billion, probably 1 billion is microarrays and the rest is sequencing. But if you look at numbers of samples, it's probably 5 million bead arrays a year and probably a few 100,000.
Well, my
sequences.
advice to you is you should be giving this talk more often, because I don't think the rest of the world fully understands that. I certainly didn't appreciate it. It's pretty remarkable.
Another question. Monica.
Maybe this is super silly and my ignorance, but you said you're measuring 28 million of these base pairs things, but there are 3 billion, and then you only do 30 times more. Is that how is so little error? I mean, because you still have to have a sample that have 3 billions of them. So how do you know with only 30 times more? I mean,
well, so the question, I think, is, you know, with only 30 times as many beads, how are you sure? And sorry to be technical, but this has to do with Poisson statistics, right? If you just run the Poisson equation, and you can be sure that if you have 29 million sequences, and I don't know what it comes out to, that at least you're going to have at least 10 of each. The minimum number of beads of any particular sequence that's going to be in there is going to be 10. So, you know, it's a huge distribution, right? You might have 60 of one and of one sequence and 10 of another, but you're going to have a statistical representation, and you know where each of the beads is. So you take the 10 that you know to be that particular sequence, and if 9 of them turn out to be an A and 1 turns out to be a G, you're pretty confident that it's an A.
I guess you cut it or whatever. Yes, I understand.
Okay. Great. Time for one more.
Hi, thanks for the talk. I'm wondering, so you mentioned that your beads that you were using were still on a micron scale. Is there any reason why you're still limiting at the micron scale rather than just down to, like, say, 100 nanometers, other than just being that you want to analyze it using visible light?
It's a great question. I mean, I think both companies have gone, certainly with sequencing, the micro-well arrays are at the nanometer scale. They're definitely at the nanometer scale, just to get the density, to get the 18 terabytes of 18 terabases. The reason that it turns out you don't want to get too small is that beads start to stick together. So you get aggregation, and the manufacturing gets to be really challenging when you get too small. So, they sort of sampled beads of all sizes, and came up with like 1 micron as being kind of the best for eliminating aggregation, keeping the density high, but not so is a compromise.
What about optical decoding? That must be the limitation too, right?
The decoding turns out to be pretty easy to solve, because what's actually done is each of the beads carries now a DNA tag, and then they use imaging probes to hybridize to these little barcodes.
Resolution limit at 100 nanometers. You're not gonna
Oh yeah, well, the pitch, yeah. So that's right. And that's another answer, is that the pitch size is optimized for the microscope, right? If you get too small, then you run into the Abbey limit. But so if you get too small of a bead, even if the pitch size is right, you don't gain anything. Yeah, good point.
Okay, well, look, this has been a remarkable story and journey you shared with us. Congrats again on the prize, and let's thank David again. Got a few comments here as we wrap up. I want to thank all of the speakers for a really fantastic day, making this new model for the symposium format a huge success. I want to thank the symposium sponsors. You've been partners with us for many years and really are critical both for letting us host a great event, but critical for connecting us to your organizations and letting the IIN have a bigger presence. I want to thank the IIN staff and volunteers. I'm going to ask that group to please stand because you've been working on this for many days and weeks. And then, thank you, I'd like to ask Deb Chandler to come up front. Deb, come on here. It's not yet. It's not because we're first going to give you something. Oh my gosh! It's a glass of red wine.
Where's my tequila?
That's. You know, I know you need that every day after working with Chad, but.
Oh my God! Thank you so much.
Deb has been part of the IIN team for 20-ish years, has been involved in this event every time, and you know, my many years here, and I know many of you in the audience just come to know Deb as one of the faces of the IIN that's really been a great partner, extremely helpful to us, and a pleasure to work with, and wish you all the best in retirement, and we'll miss you.
Thank you. Thank you. It's been my pleasure.
Fantastic. Now, I think there's a reception out in the common area there, and ask everybody to stay around and mingle.